Okay, I'll go ahead and get started with the introduction. Thank you everyone for joining us today for this webinar with North Central Climate Collaborative. We appreciate everyone being on. Today's speaker is Dr. Christine Sprunger. She's an assistant professor of soil health in the Department of Plant, Soil, and Microbial Sciences, which is based at the Kellogg Biological Station in Michigan. Her research focuses on the intersection of agriculture and the environment, and she investigates how various management practices impact soil health and ecosystem services. In addition, Dr. Sprunger is interested in understanding how nematode communities can serve as a key soil biological health indicator within agroecosystems. And lastly, Dr. Sprunger also likes to explore how climate change impacts rhizosphere dynamics and soil food webs. Today, she'll be presenting her talk on enhancing soil health, opportunities for climate mitigation and adaptation in agroecosystems. Before we get started, if you're unfamiliar with the webinar format, if you have a question for Dr. Sprunger, which she'll take at the end of her talk, you can either put it into the chat box and we will be monitoring that, or you can submit it as a Q&A, which those buttons should be along the bottom of your screen. And so you can just do that and she'll take time for questions at the end. And with that, here's Dr. Sprunger.
Great, thank you so much. And thank you everyone for tuning in. So I probably don't have to tell this group that there's an increased interest in soil health. And just so we're all on the same page, the definition that I tend to use when thinking about soil health is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. And one of the reasons why soil health has gained such widespread attention, not only from farmers and scientists and extension educators, is that it just leads to a more holistic approach to testing our soil. So really key components of soil health is thinking about biological processes, physical processes, as well as chemical processes. And generally when we think of traditional ways of soil testing, the indicators that are available to farmers tend to just be that chemical component. So soil health, again, just provides a more holistic view of our soils, which we think of as really important for understanding key ecological functioning in our agroecosystems. So, for example, it is assumed that increases in soil health could lead to greater biological diversity. We know that other benefits can also be related to crop productivity and water benefits, such as better water quantity and water quality. And then lastly, thinking about carbon storage. And a large part of this talk is going to be thinking about how soil health can be used to sequester carbon, which is critical for mitigating climate change. The other really important thing about soil health, again, is that it is really important at local and regional scales. So when I talk to farmers in Ohio and Michigan, they're often asking for more information from their current soil testing. So again, they want more information than just kind of fertility information. They want to understand the biological aspects of their soil and also the physical components of their soil. There's also enthusiasm for tests that are more sensitive to management. So for example, a farmer may get a test result back from a commercial lab that looks at percent organic matter. This percent organic matter can often take decades to see changes, even when soil health promoting practices have been incorporated. And so part of the kind of soil health framework is coming up with indicators that are more sensitive to management that can help farmers understand where they are in terms of kind of their trajectory in terms of improving overall soil health. When I talk to farmers, especially in Ohio, there also seems to be kind of a pressure to improve soil health from a water quality standpoint. And so, of course, there is also interest in linking soil health to nutrient retention, especially when thinking about like water qualities in Lake Erie. And then lastly, I think there's just a huge opportunity to understand how soil health plays a role in climate mitigation and adaptation, which again is the main focus of this talk today. When we think about kind of opportunities for climate mitigation within agroecosystems, we have to first consider soil carbon. So we know that soil carbon and soil health are intricately linked. And historically, soil carbon has really been diminished within our cropping systems due to intensive management practices. So locally, 40 to 60% of original carbon has been lost within North America. And this map down at the bottom of the slide demonstrates, the darker the color, the more carbon has been lost since early pre-settlement. And so with this loss in soil carbon also creates an opportunity to rebuild these carbon pools. And once, we increase these soil carbon pools, that also leads to carbon sequestration and opportunities for climate mitigation. But how do we actually do that in practice? And so one of the ways in which we can try to increase soil quality and soil health and soil carbon, all these important things, are to incorporate soil health promoting practices. So when I think about soil health promoting practices, I think of kind of 4 key principles. The first is just to increase organic matter input. So that could be incorporating cover crops that have deep roots that when they turn over are contributing to carbon and overall carbon sequestration. It's also important to diversify soil biota with plant diversity. So just lengthening your crop rotation, not just having corn and soybeans, but also thinking about wheat and other small grains that can be incorporated into the rotation. It's also really important to think about year-round ground cover. So again, having maybe a perennial or a cover crop. So just reducing your soil's exposure to erosion. And then lastly, it's really important to think of living roots in the ground throughout the year. So again, that can be achieved through either incorporating perennials or cover crops. But a question that we need to ask is, do soil health promoting practices actually increase soil biological health? And the first story I'm going to tell you today works to address this using a biodiversity gradient. So this is a long-term trial located at the Kellogg Biological Station. It's primarily a biofuel cropping system experiment. But what I really like about this is we can compare things on the lower diversity end, just looking at continuous corn and then moving into corn and soybeans with cover crops, and then moving into our monoculture perennials such as switchgrass, miscanthus, and poplar, and then moving into our more diverse rotations of native grasses, early successional, and restored prairie. And actually, before I go on to the next slide, it's important to note that that poplar system, it is traditionally thought of as a monoculture perennial, but it actually has a vigorous understory. And over time, we've realized that actually we should also be considering this a polyculture system. We also have to think about how we're actually measuring and quantifying soil biological health. So the indicators here are really key in reflecting soil carbon cycling and carbon sequestration. So many of you might have heard of respiration or mineralizable carbon. This is a 24-hour assay that you're just measuring CO2 coming off of your soils. And this is critical in really understanding the most labile pool of carbon. So this is the carbon that's most readily available to your microbial community. And it's going to be the most sensitive and reflective, most sensitive to recent changes in management. Then you have an active carbon pool. This is also known as permanganate oxidizable carbon, and this is measured through a chemical reaction. And active carbon also is measuring a pretty labile pool of carbon. But again, over time, what we've noticed is that it actually reflects a slightly more processed pool of carbon and can actually serve as an important indicator of carbon stabilization. And then lastly is soil protein. This is a labile pool of nitrogen. It is highly correlated with soil organic matter cycling and is also just a really kind of new innovative indicator that reflects overall nitrogen cycling. So what did we find? So the way to read these graphs, in gray, we have our annual cropping systems. In orange, we have our monoculture perennial systems. And in gold, we have our polyculture perennial systems. So for graph A, we're looking at that respiration indicator or mineralizable carbon. And we see quite a magnitude difference between our polyculture perennials, monoculture perennials, and annuals, where the annuals have significantly lower mineralization rates relative to the monoculture perennials and the polyculture perennials. When we look at graph B, we still see differences across the gradient, but the magnitude is much reduced. And what I really like about showing these graphs side by side is it really demonstrates that mineralizable carbon and POCs are reflecting functionally different pools. Again, mineralizable carbon is just a much more sensitive indicator. POCs is still sensitive. It's still detecting differences across these different systems. But because it's reflecting a more processed pool, it does take more time likely to determine and detect these differences. I also failed to mention that when we sampled in this study, these systems had been in place for 9 years. So again, that kind of shows you the timeline of what you can expect to see in terms of carbon accumulation over time. When we look at the soil protein, again, this is reflecting labile nitrogen. It pretty much tracks with the respiration value. So again, we see just much lower levels within the annual systems. So just some quick kind of implications of the study. Systems with greater perenniality and crop diversity increase soil biological health. Perennial polycultures, aside from restored prairie, increase active carbon relative to annuals, which has really important implications for carbon stabilization. And it's also important to note that even within the annual systems, rotated annuals with a cover crop increase active carbon relative to continuous corn. So even if you are only working with annual row crops, including a cover crop is really important for accumulating carbon. And then lastly, these soil biological health indicators have the potential to serve as indicators of both nutrient availability and soil carbon stability. So that's climate mitigation. The next thing we need to think about is climate extremes and adaptation. So I likely, again, don't have to tell this group that the Midwest is definitely experiencing climate change. What we've seen over time is a steady increase in precipitation, and this falls in terms of intensive rainfall events followed by longer periods of drought. So just overall, more extreme weather events are occurring, leading to just a greater overall precipitation. What we've also seen over time from the literature is that because of climate change, yields are becoming more stable, and the way to stabilize these yields is to incorporate crop diversification and improve overall soil carbon. For example, there was a study by Dr. Dan Kane that demonstrated that even under drought, increased soil organic matter is really what protects maize yields from reducing due to variable weather. What hasn't fully been addressed is how does drought impact soil health, especially within a single growing season? So how likely is a drought event to actually reduce soil health and impact soil health, which could have a negative impact on yield? And that's what we'll talk about next. So the research aims for this next study were to examine how long-term management impact soil health and soil carbon stability across a management intensity and plant biodiversity gradient, and also assess how sensitive soil health indicators change temporally over the course of a single growing season in response to recent weather patterns. So this study was taking place again at the Kellogg Biological Station, but this time within the National Science Foundation Long-Term Ecological Research Site. This trial was established in 1989 and is a management intensity gradient that looks at annual, perennial, and unmanaged systems. So the treatment kind of key for this study is T1 through T8, and in your T1s through T4s, these are corn, soybean, and wheat rotations that are managed under conventional, no-till, and reduced input. Reduced input is just the same as conventional, but it receives 1/3 of the fertilizer and it's grown with a cover crop. And then biologically based doesn't receive any external inputs. It's just getting its nitrogen from a cover crop. And then you move into your perennial system, so poplar and switchgrass, and then you move into your polyculture systems, the early successional community, mown grassland that's never been tilled. And then this is just to note, this is a change in precipitation here at KBS from our KBS weather stations. And again, this is the same trend that we're seeing across the Midwest, just a steady increase in precipitation over time. But again, you can see kind of that flashiness as well. So we also see prolonged droughts. So the way that we're assessing soil health in this study is very similar. We're going to focus on respiration and active carbon again, but this time rather than just focusing on one sampling time point, we actually sampled at 4 times over the course of the growing season. And again, just a reminder that respiration and active carbon are reflecting slightly different pools of carbon. And it's really important to actually measure both of these because they can inform, again, this carbon stability. So many of you might recall that in 2021, we had a pretty substantial drought at the beginning of the growing season. This was one of the largest droughts on record for the early part of the growing season. And it was very reminiscent of 2012, for those of you who remember that drought year. But we finally got a lot of rain in mid-June. And so just keep this graph kind of at the back of your mind as I walk through some of these data slides. So again, the way to read these graphs, I've kind of separated these out by annual crops, the monoculture perennials, and then the unmanaged systems. And right off the bat, we clearly see a management difference where the unmanaged system has greater overall mineralizable carbon relative to the annual crops and the perennial crops. We also see a lot of variability here with the large error bars, and that's one very emblematic of respiration. It tends to be quite variable. And 2, I think it also kind of reflects to the mechanisms that could be occurring here. So one thing that I want to draw your attention to, especially in the perennial and unmanaged systems, is that there's this big drop in respiration between May and June. And again, what we are hypothesizing that's happened here is that it's due to the drought. And what happens when you have drought is you have these dry conditions where you have reduced microbial activity, and it isn't until you start to have a re-wetting event or an actual precipitation event where microbial activity increases and then they start to respire more. And that's where you can kind of see that upswing in respiration from June to August. We actually see pretty similar trends in this active carbon value. One thing I'll note is that in the unmanaged system, it's the mown grassland, that dark red figure line is slightly higher and actually a little bit more stable, especially between June and August. And that's really important from a carbon stabilization standpoint. Again, this is a polyculture system, likely has deep roots. We know it has the most amount of carbon, even when we think of just total carbon. And so again, even though it's experienced the same amount of drought, it still kind of can recover from that drought, I would say, a little bit more quickly than the other systems. And again, you do see that drop in active carbon between May and June. So I think that the same kind of mechanisms are occurring here as what we saw for respiration. Okay, so this next graph we'll walk through a little more slowly. So again, what I like about measuring both respiration and POX is that we can actually conduct a linear regression model and really understand the relationship between the two. And based on that model, we can extract the residuals. And the only thing that you really need to take away from this is that a positive value means that the system is actually more controlled or more influenced by stabilization processes, and a negative value is more influenced by mineralization processes. And so another way to say that is a positive value means that the system is actually more influenced by POX or active carbon, while a negative value is more influenced by respiration. And this is a really important kind of finding because it really demonstrates how a system might be in terms of its overall carbon trajectory and in terms of its ability to really stabilize carbon over time. So one thing that really jumps out to me is the annual crops. We see that the majority of those bars are negative and they're kind of trending towards mineralizable carbon, which could mean that they just aren't as efficient at kind of accumulating and stabilizing carbon. Whereas the perennial and unmanaged systems have more positive bars. The only system to have 4 positive bars over the course of the entire growing season is that mown grassland. So again, that is the system that seems to have the most stable carbon, even with experiencing a drought in the beginning of the year. So just a study summary here. Active carbon and respiration are really key soil health indicators that fluctuate temporally and are sensitive to not only management intensity, but changes in precipitation. This demonstrates the role that soil health indicators can play in detecting system level responses to climate extremes, and soil carbon appears to be more stable in the perennial and unmanaged systems relative to annual row cropped systems. The other thing we need to think about is that, we're not just experiencing drought. We are also experiencing early summer flooding events. And this has implications for soil biological health. So this figure on the right demonstrated with an increase in precipitation and early summer flooding events, you see a decrease in suitable workable field days. So this demonstrates that it's really hard. It's becoming harder for farmers to find times to plant, times to work their field early in the growing season. And that's likely due to these intensive flooding events that we're seeing more frequently. So this next study that I'm going to talk about is the USDA NIFA funded project that works to assess flooding impacts on soil health and crop productivity. So this is a grant that I received with one of my colleagues, Dr. Alex Lindsay at Ohio State University, and his lab is focusing on kind of the agronomic performance piece, while my lab focuses on the soil health and kind of below ground rhizosphere dynamics piece. So this study has two sites. The first site is located at the Northwest Agricultural Research Station, and then the next site is at the OARDC in Worcester. And the kind of three main goals of this study are to, one, assess the intensity of flooding on soil health and crop production, and two, determine the impact that nitrogen source and rate have on above and below ground production, as well as soil health post flooding, and then also assess how different cover crops impact water cycling, soil health, and crop yields in the event of flooding. So today, I'm really going to focus on the nitrogen source kind of aspect of this. And so the way that these trials are set up is, we have these large scale trials. And to simplify the trial for today, what we're going to focus on is kind of soil health in terms of being sampled in the pre-flood, in a system that's received urea, and a system that's received manure. And then a post-flood, thinking about urea versus manure. So in this study, I'm actually going to talk about a different aspect of soil health. So one thing that really strikes me in the soil health framework is we often are relying on things like respiration and active carbon to reflect biological processes, but we aren't directly measuring soil biota. And I think that really needs to change in terms of, helping soil health actually address biodiversity issues. And one thing that has also motivated me to think about this a little bit more is that farmers often ask me for soil health indicators that directly measure soil organisms. So the way that we kind of are working to address this is thinking about nematode communities as indicators of our overall soil health. And for just to make sure that everyone's on the same page, the reason why we're interested in nematodes is because of their kind of strategic placement within the soil food web. So nematodes are microscopic, non-segmented worms, and oftentimes people are like, well, how large is a nematode? And you can often think of the size of a nematode as 1/500th of an inch. So very microscopic. We have to use a microscope to count and identify them. So nematodes are really important within the soil food web because they're specialists. And so there are nematodes that only feed on fungi, nematodes that only feed on bacteria, nematodes that are plant parasitic, so those are the ones that feed on plant roots. And then there are nematodes that are predators, they actually feed on each other. And then there are nematodes that are omnivores, meaning that when they, in any given kind of environment, they can kind of change their feeding preferences. And then based on the ratio of these different type of nematode to nematodes, we can actually learn a lot about overall soil biodiversity and ecosystem health, and also nutrient cycling. So we just think that this is a great link to soil health and ecological function. So the way that we sample nematode communities is that we take a soil sample and then extract them using Bierman funnels. This is my grad student, Tavisha Martin here. You can see she has a series of nematode extractions that run over a 72-hour period. And then she just kind of waters them once a day, and the nematodes kind of swim to the bottom of the funnels, and then she releases the clamp on that tube and then collects them after 72 hours. And then she counts and identifies them using a microscope. So we wanted to just see, this is kind of hot off the press data, but we just wanted to look at nematode abundances across these kind of pre and post flooding events. And what we see here is that regardless of the nitrogen source, there's a decrease in nematode abundance after a major flooding event. So in this case, there was a 4-day flooding event. And we see, especially within that manure nitrogen source, we have a substantial decrease in nematode abundance. And then we also see a decrease in nematode abundance within the urea, just not to the same extent that we do in the manure. Also important to note, though, is that even though we see that substantial decrease in nematode abundance within the manure, it's really just on par with the pre-flood nematode abundance within the urea. So this is telling me, one, that manure additions really increase the overall abundance of nematodes, and even after a flood, their nematode communities can be maintained when you have an organic source of nitrogen. So again, this is just kind of a preliminary data that we're working with here, but it's definitely clear that nematodes appear to be really sensitive to flooding, and significantly and substantially reduce their numbers after a flood. So the next step is to identify them. So again, I said this is kind of hot off the press data. My technician was just finished counting these on Friday for me. And so the next step will be to identify them. And the reason why we want to identify them down to feeding group and even family is so that we can calculate these nematode indices. And again, this is why we think of nematodes as being critical for soil health, because we think that they can really reflect important ecological functions. So I'm just going to walk through these just to give you an idea of what we'll hopefully be able to learn in this study. The structure index is just an overall trophic complexity and diversity. So oftentimes the structure index goes up when we see more predators and omnivores. So this is kind of like the larger nematodes that are really having a top-down impact on just the nematode community. And then we have the enrichment index, and this reflects an increased organic input or increased nutrient cycling. And so again, I'm really interested in seeing how that enrichment index responds within that manure versus urea treatment. I think that will be a really important index to keep our eye on. Then we can also look at the channel index. This is looking at fungal to bacterial decomposition pathways. This gets important again when we think about soil carbon stability and thinking about fungal versus bacterial pathways. Oftentimes when we see a system trending more towards fungal decomposition pathways, that can reflect more carbon stabilization. And then lastly, the basal index reflects the amount of disturbance. So again, this is going to be really important to understand within the context of this flooding project, and also just in general for climatic extremes, because it can be an early indicator of disturbance and reflect overall ecosystem health. A way that we can actually put these nematode indices into practice is just focusing on the enrichment index versus the structure index. So this is the left figure is adapted from Howard Ferris in 2001. And you can see that you have this quadrant where, when you have a high enrichment and a low structure index, you have a very kind of disturbed system. When you have a high enrichment index and a high structure index, you have a more maturing system. And then when you have a low enrichment index and a low structure index, you have a degraded system. And then when you have a low enrichment but a high structure index, you have a more matured fertile system. So that's oftentimes we see that in forest systems. So you're not getting that high inorganic N input, but you still have a healthy kind of overall nutrient cycling. And so we tested this in another study that was conducted in Michigan, comparing annual and perennial cropping systems that received organic nitrogen and inorganic nitrogen. And what you can see here is that the squares reflect that organic nitrogen. So both systems, regardless of if it was annual or perennial, are in that more maturing nitrogen enriched kind of quadrat. And then the annual wheat system that received inorganic nitrogen is in that degraded kind of square. So this is just an example of why we think these nematode indices are useful, and a really strong link to soil health. So lastly, I just want to briefly mention another drought study that we're working on at the KBS LTER. We're actually we just finished our second year and we'll have one last final season next year. But we constructed numerous rainout shelters to explore drought. This is part of a large study. There are kind of 9 different labs that are looking at various aspects, from everything from plant communities to pollinators to biogeochemistry. Our lab is, of course, focused on soil health and nematode communities. But it's, again, I wanted to share this to demonstrate that a lot of people are working to understand drought impacts within agroecosystems, and we look forward to sharing those results with the broader scientific community soon. So with that, I just want to give a final summary. Crop diversification and perenniality appear to be effective at carbon sequestration, which is critical for climate mitigation. Drought negatively impacts soil health, but crop diversity and perenniality can aid with climate adaptation and carbon stability. Flooding significantly decreased nematode abundance, which could be an early sign of decreased soil health. And lastly, organic sources of nitrogen could help to maintain nematode communities during flooding conditions. And so again, that just reflects and relates to that climate adaptation piece and what farmers could think about doing in terms of nitrogen management, even when experiencing these extreme weather events. And lastly, I just want to acknowledge my funding sources and my lab, because they're the ones in the field and the lab doing a lot of this work. So thank you all for listening, and happy to take any questions.
Thank you, Christine. That was great. We have some questions here in our Q&A, so I'll read those. Everyone should be able to see them, and then you can answer. The first one came towards the beginning of your presentation, and they ask, have you had an opportunity to share your research with farmers and landowners? How open are folks to these soil health practices?
Yeah, so that's a really great question. We've shared a lot of these soil health indicators with farmers, not from the context of sharing these long-term trials, but actually going on farm and sampling different farmer fields, and then generating soil health test reports and actually interviewing farmers after they've consumed that information. And what we find is that farmers are actually very receptive to these soil biological health indicators. Actually, one of the questions that we often ask is, which indicator seemed to be most useful to you? And in one of our studies that was working with Ohio farmers, we found that the majority of farmers actually named a specific soil biological health indicator, especially that respiration and pox indicator. And I think one of the things that really connects with them is that these indicators are able to detect differences across fields that they're very familiar with. And I think that to them demonstrates that it's a useful tool. And so, yeah, I can chat a little bit more about that if people are interested.
Okay, great. The next question was, adding cover crops seems to decrease carbon mineralization compared to corn and corn soybean. Why is that?
Yeah, let me just go to that bigger. Yeah, so this is a really good example of when you have corn versus like corn and soybean in rotation versus a continuous corn and the corn plus cover crop. So it does slightly decrease it. It's not significantly different. But I think what I would say here is that lengthening your crop rotation, having the corn, soybean plus cover crop is likely the better route to go than just always having corn in your rotation. So Yeah, I'd have to think a little bit more about why we see that decrease. But again, it's just a slight decrease and it's not a significantly different drop.
Sticking with the cover crops, the next question was there a diverse mixture of cover crop species, or did you use a single species? And were tillage practices looked at as a variable?
Yeah, so great question. Just a single crop species. This was just a clover. And these were actually all no-till systems. Because again, we're comparing against perennials, and so they wanted to keep it no-till.
Has there been any analysis of microbial communities and how they fluctuate?
In this specific study, there have been a couple different studies. So for example, Lisa Tiemann, she looked at the continuous corn, switchgrass, and the restored prairie, and it follows the same trend where you have an increased overall soil biodiversity in the polyculture perennial systems relative to the monoculture and annual system.
Another question you said that adding manure to a soil can maintain nematode populations. Would non-manure compost have the same effect?
You know, that's a really good question, and that's something that I haven't personally tested, but I would hypothesize that yes, we'd likely see that same effect. I think it's really that organic source of nitrogen, that enriched organic matter input relative to that inorganic nitrogen source. So yeah, I would hypothesize that you'd likely see that same response.
Great. What major factors do we need to see when selecting cover crops and multi-species cover crop to manage soil health and regenerative ag practices from small producers, like from the initiative perspective?
Yeah, so I can think back to another study that we actually did on farm, and we saw consistently that in either lengthening your rotation with either a cover crop or incorporating some type of perennial grass, whether it be alfalfa or any other kind of grass where you're that's in your rotation for more than a year is going to be really beneficial for improving especially soil respiration and pox. And so again, it really gets back to those soil health promoting practices and principles of lengthening your rotation and making sure you have roots year round. I would love to do a more intensive study looking at a variety of different cover crops, and we actually have that as part of our flooding project. So we'll be able to kind of shed light on the effectiveness of monoculture versus mixed cover crops, not only on soil health, but just overall maintaining soil health even in the event of a flood. So please stay tuned. Hopefully in the next year, we'll have data to share on that.
Adele Durfee just says great presentation. So not a question there, but thank you, Adele. Another question. I haven't looked into the cost profiles for the pox, carbon, and nematode testing. Is this something that is, or hopefully could be in the future, a cost-effective approach?
Yeah, so another really great question. What we have calculated in our own lab is that respiration and pox as a package can be pretty affordable. We can offer it at about $6 per sample. Nematodes are slightly more expensive simply because it takes a lot of labor to count and identify. And so I would love to ramp up our capacity. And a lot of people ask about, well, are there molecular methods that you can use versus just counting? Unfortunately, the primers needed to detect nematodes are just not there. And right now, molecular methods only have about a 30% accuracy of accurately identifying a nematode. So we still have to kind of stick with the microscopic approach, which just takes a lot of time, which is why I didn't have that data to share with you today.
Great. Next question. Has the work involving the farmer interviews and farmer interest in specific indicators been published?
No, we are hoping to submit that by the end of the month.
There you go. Stay tuned.
Yes.
Perennial systems improve soil health, but also seem to produce less food. Do you have thoughts on that?
Yeah, that's a really important thing to consider. So the last graphic that I kind of shared is focused on perennial grain crops. So I'm sure some people are familiar with the Land Institute and breeding, perennial grains as a way to not only provide these ecosystem services, but also get grain yield that could actually feed people, other than, the first story was just focusing on these biofuel cropping systems, which aren't going to feed people. There is no easy answer to this other than these perennial currents. Crops are not quite ready for the commercial market. We are working with some farmers in Ohio that are kind of experimenting with perennial grains. But again, you're right. That's kind of the conundrum and trade-off, is that while these perennials are effective at ecosystem services, they don't at this time have the capacity to meet food security issues. So the likely kind of best approach then is to think about how we can increase cover crops and lengthening our rotations and focusing on just overall enhancement of soil health within annual row crops.
Great. We still have a few more questions. Are we good on time for you? We're still under 2. It's not 2 o'clock yet. So, all right, we'll keep going. Do you work with any farmer-led groups? Farmers often listen to other farmers more than they'll listen to a scientist. They're doing some great, can do some great work in this regard.
Yeah, so one of the really exciting new projects at the Kellogg Biological Station is we have a new long-term agroecosystems trial. So the LTAR network, and this is all stakeholder driven. So we have a stakeholder advisory board that actually helped design our business as usual and aspirational treatments. And we're hoping to have kind of more outreach events with these stakeholder advisory boards that are mainly farmers. And because you're right, I think that we need farmers to kind of spread the word about soil health promoting practices and overall, soil health. So one example of this is we actually are really interested in prairie strips as a rapid soil health indicator. And we have a couple of farmers here in Michigan that are experimenting with prairie strips, and then they have demonstration trials or demonstration kind of field days on their own farms. And they have been the most effective at getting farmers to participate with and experiment with prairie strips. So totally agree.
Great. Okay, next question. And while we're doing these questions for the attendees that are still on, which is still quite a few, I'm going to do a couple poll questions. So if you could answer those as we're going through the end of the presentation here, that would be great for our metrics. Next question is there any evidence that increasing the POX, carbon, and the mineralizable residual number, is it actually linked to carbon sequestration in the long term?
Yeah, so that's something we're testing in the KBS LTER. And the numbers that I shared with you today, perfectly align with the soil carbon sequestration values that we've consistently seen in these long-term trials at KBS. So that's why we're so excited about these indicators as being effective tools for early detection of carbon sequestration. I will say that we have another new funded project that's through the Environmental Defense Fund, and we're actually wanting to test the accuracy and ability of these indicators to predict carbon sequestration, not just at KBS, but across sites, all across the United States. And so what we're doing is we're using archived soil samples. Going back 20, even 50 years across all these long-term sites and measuring POX and respiration, because you can measure these on dried soils. And then looking to see, which indicator is actually most accurate in terms of predicting future, present day carbon sequestration kind of values. And the reason why the Environmental Defense Fund funded this is because they want to really understand which indicators might be used to quantify soil carbon accumulation on farm, which could also inform carbon credits.
Great. Which soil carbon measurement of the two might be best to use in grazed pasture?
Yeah, I think I'll stick with I think we need to measure them both. But if you had to pick one, likely the one that's going to be most accurate and more stable over time is the active carbon or permanganate oxidizable carbon. Respiration is great in terms of, if you have two fields that you really want to understand different management practices and detecting differences between the two. It's really good at picking up those differences, but it's just more highly variable. So I think a more cautious but better accurate measurement is probably that POX value.
Great. Next question. Trying to squeeze the rest of these in here. The practice of retaining winter high flows on ag fields and orchards as a means of groundwater recharge is increasing. What should practitioners be aware of to restore nematode or other soil biota?
Yeah, that's a really important question. Again, I think it's having living roots and year-round and having year-round ground cover, whether that be through having a perennial in your rotation or a winter cover crop. I just think that reducing, minimizing your soil's exposure to the elements and reducing overall erosion is really what's going to maintain your soil biota.
I'm going to squeeze in a quick question really quick.
Sure.
I'm curious, what depth did you sample for your nematode population?
Yeah, so all of the data that I shared today, we went to 10 centimeters.
10 centimeters, okay.
Yeah. Mm-hmm.
I've done some nematode extractions in my day, and so I was kind of curious how far you went.
How far did you go?
Oh, I think it was about the top 3 inches. So yeah, about the same. So thanks. Okay, moving on to the next question. What about daikon radishes as a cover crop with clover?
Ooh. Yeah, I'm not, I would say, an expert at like mixed cover crop species. I can only point to what I've seen in the literature, and generally what we see is, if you can increase your overall cover crop, like the number of cover crops that are grown together, you generally see greater ecosystem services. But yeah, I can't really speak to that specific combo.
Okay, another question. Fantastic talk. Anthony's intrigued by your frame of using mineralizable carbon to index the carbon instability and the POX carbon to indicate carbon stability. But that carbon stability graph you showed suggests there's a lot of flashiness, maybe enough to freak a producer out. How close do you think we are to a more stable and robust index of stable soil organic carbon?
Yeah, no, that's a really important question. And I think, as scientists, we're always working to kind of search for that next indicator. I don't think there's ever going to be a perfect indicator, which is why it's important for us to use, several indicators, several different tools to try to get at the question. So some people are really interested in enzymes. We also did enzymes for that same study, and it was even more variable than mineralizable carbon. So I would say, like, if I had to pick one, it would probably be that permanganate oxidizable carbon. But yeah, I think I still like to do both when I can.
Okay. We got about 5 more minutes. Let's see what we can get done here. How do you try to encourage farmers to make these practice changes?
I like to be able to show the data. I like to empower farmers to make their own decisions, but giving them tools that are actually going to be more informative. So again, when we sit down and have these farmer interviews, we're able to give them results from what they would get at a commercial testing lab, and then these soil health measurements, so they can see for themselves which indicators are most useful. And a lot of farmers will say, if I just relied on that % organic matter value, that is not changing even after I've done no-till for 10 years. Why do I continue to implement these sustainable management practices? But then when they see these more sensitive indicators of soil health that are demonstrating a magnitude difference between their different fields, that gives them agency to know that they're on the right track.
Great. Is there research with full inputs on no-till with cover crops?
Oh, yeah. I am not a part of any of that research, other than, these long-term trials that are working. The first study that I showed, that's a no-till system where we have cover crops within that annual rotation. But in terms of like on-farm trials or larger kind of strip trials, I'm not a part of that work, but I know that that's something that a lot of people are very interested in and focused on.
Great. Are there any general thoughts or speculation regarding the addition of livestock, especially grazers, into the systems and rotations?
Yeah, so it's another really great question, and it's something that we've thought a lot about within this new LTAR site at KBS. Again, these are, large plots relative to what we see in most kind of, on station trial studies, but they aren't quite large enough to incorporate actual livestock. So right now what we're doing is just using manure to kind of represent that animal component. But there is talk of using our field scale up fields to try to incorporate some type of grazing component, because from the literature, we do know that livestock integration with row crops could likely be the most sustainable option. It could be really critical for improving overall soil health.
Yes. Has KBS been collecting any type of hydrological function properties with soil, like infiltration rates, aggregate stability?
Yeah, so we've done a little bit of aggregate stability work. Again, we just have really preliminary data for that, so I can't speak to that today, but there are a lot of people that have looked at kind of hydraulic cycling, especially in the perennial biofuels. And again, very similar trends in terms of the perennials just being overall more efficient in their cycling. And this also leads to greater loss of nutrients. So there have been studies that have looked at both nitrate and phosphorus leaching, and those perennial systems are just really effective at retaining those nutrients.
Great. There's still a few more comments here. We're approaching almost 2 o'clock. I'd like to call everyone's attention to the chat. I put in our North Central Climate website link to our webinars page, and in a couple days we will, or maybe sooner, depending, we will be posting this recording of Dr. Sprunger's talk, so it'll be available to you there. And I would imagine that she would welcome any follow-up questions to her email, which I can put in the chat. I can pull up my window here. But I guess there's a couple more on here. I don't know if we have time, as we're trying to keep to an hour, but I don't know if Christine, if you can type your email into the chat, that might be helpful, because I've got a lot of windows popping up over here.
Please feel free to email me with any questions you might have.
I appreciate everyone being on. We had a really good attendance, and I thought this was a wonderful presentation. Thank you so much.
Thank you.
Great. All right. I think we'll call today done, and we will have another webinar in 2 months. Be on the lookout in your email for more information about our next NC3 webinar. And thank you very much for joining us.